What's up, everybody? Let's take a minute here to talk about your expectations for the week, your assignments, and then at the end, we're going to talk a little bit about the cell cycle and cell division, mitosis. So you'll have some background information while you're working on these things. So let's go ahead and get started. This week, there's not going to be a formal Zoom meeting. Just make sure you watch the video and then go to the check-in Google form in our Google Classroom and fill that out. Let me know if you need anything from me. If you need to Zoom with me, we will totally set that up because I want to help you as best as possible. And otherwise, just send me some emails for questions you might have and do the check-in. Monday, there is an Edpuzzle video where you look at the cell cycle, go through and slowly and carefully watch the video and answer the questions. Make sure you take your time because, as you know, Edpuzzles only let you answer once, so take your time, answer accurately. Tuesday, there's a second Edpuzzle. Monday, the one is about the cell cycle, like an overview. Tuesday is specifically about mitosis. So do the same thing. Watch the Edpuzzle, take your time, be accurate. And then Wednesday, for the rest of the week, there's some questions that I want you to... There's a Google Doc where you're going to read... it's like one page. You're going to read one page, and then you're going to observe this diagram, look at the parts of the diagram, and then there's some questions at the end of it. And all you really need is the document itself, the information and the diagram in the document itself. But I have attached that chapter of your textbook just for extra backup information. And as you all know, Google is out there too to help you find stuff. But the document itself should be enough to answer the questions in the document. And I'm going to give you the rest of the week to turn that in. So just give me those 3 things this week and you will be good to go. If anyone needs to go back and turn in some makeup work, make sure you're doing that. I don't want none of this stuff to pile up on you by the end of the semester and end of the quarter and make it difficult to pass. I want you guys to be thinking about this smartly and get yourself in a good place because I want you all passing. Okay, so let's go ahead and shift gears here and talk about the cell cycle and mitosis. We are going to look at how cells grow and how they reproduce. So that's what we're going to focus on this week. There's a few more things we're going to learn about with these processes in the near future, but this is what we're going to specifically look at today. So cells have a size limitation, and the size of the cell influences whether it needs to reproduce or not, and we'll get into that in a second. So several factors influence a cell's size. Surface area is the area covered by the plasma membrane, and the volume is the space taken up by the inner contents of the cell. And cells with a higher surface area, or a surface area to volume ratio, can sustain themselves more easily. Now, when you look at these pictures, you can see the nucleus is very close to the outside of the plasma membrane here. The second one, the nucleus, it's still fairly close. The third one, the nucleus is pretty distant from the plasma membrane. Now, think about what you already know about cells and how things travel inside and out of cells. Things go from the nucleus to the plasma membrane through the cytoplasm. The closer the plasma membrane is to the nucleus, the easier it is for things to transport in and out of it. The further things are, the plasma membrane is from the nucleus, the more difficult it is for things to travel in and out. So at a certain point, cells get too big where they only have 2 choices. They either have to die, which cells don't want to do that, or divide. So once they get too big, they're either going to die or divide. And so let's look at what that looks like. Smaller cells, as I just said, transport substances more easily. Diffusion is not that efficient over long distances. The cytoskeleton is less efficient when the cells are bigger. Cellular communication is more efficient in small cells. If things are closer together, it's easier for them to communicate. So once a cell reaches that size limit, it either has to stop growing, aka die, or divide. Cells reproduce by a cycle of growing and dividing, and we call that the cell cycle. Again, they're constantly growing and dividing over and over and over. Each time a cell goes through one complete cycle, it becomes two cells. So in this cell cycle, there are three stages. We have interphase, mitosis, and cytokinesis. Interphase is the stage where the cell grows, carries out most of its normal cellular functions, replicates its DNA. Mitosis is when the cell's nucleus and nuclear material divide. It has four little substages. And then cytokinesis is where the cytoplasm divides at the end of mitosis, giving us two brand new cells from the one cell. So let's look at that. Let's look at the cell cycle. The stages of interphase, which is the first part of the cycle, has three substages. We call those G1, S, and G2. And you can see them on this chart here, G1, S, and G2. Now this chart shows the whole cell cycle, and as you can see, interphase is the majority of a cell's cycle or a cell's life. So interphase is what a cell's typically going to be found undergoing. It takes up most of the cell's life. Okay, so G1, S, and G2 is interphase. During G1, the cell is growing and carrying out normal functions. Okay. During the S phase, and I should have said G1 is called the gap phase. S is the synthesis phase. Now, during the S phase, think about the word synthesis. What does that mean? Something's being synthesized or made. During the S phase, the cell copies DNA to prepare for cell division. So chromosomes are the structures that contain the genetic material, and chromatin is relaxed form of DNA in the cell's nucleus. So again, during the S phase, DNA is duplicated. During the G2 phase, this is right after the S phase, and it's when the cell is preparing for the division of the nucleus to begin. So G1, S, and G2, that's interphase. We're about to jump into mitosis and cytokinesis. During mitosis, a cell's nuclear material divides and separates into opposite sides of the cell. And then during cytokinesis, your cell turns into two daughter cells. Daughter cells is the word we use for a brand new cell. Think of the original cell like a parent, and now it has two daughter cells. Okay, that's an overall overview of the cell cycle, but let's get into mitosis and the actual parts of mitosis. Now, we've talked about prokaryotic cells and eukaryotic cells. In eukaryotic cells, which is, our cells, animal cells, plant cells, cells divide from that cell cycle. In a prokaryotic cell, things like bacteria and amoeba, they divide by binary fission. They just kind of split apart. It's a way simpler process. So we're not even going to really focus on that too much. But eukaryotic cell division is what we are going to focus on. Specifically, we're going to look at mitosis. During mitosis, the cell's replicated genetic material separates, and the cell prepares to split into two cells. And the key activity of mitosis is the accurate separation of the cell's replicated DNA. So there's a lot of checking and rechecking in there of the DNA to make sure it's accurate and put together the way it's supposed to before it actually splits off. Mitosis, so it's going to increase the number of cells in a multicellular organism as it grows. It's going to replace damaged cells. So your cells in your body are constantly replacing themselves. And if they ever get damaged or hurt, they quickly replace themselves. And that's the point of mitosis, to duplicate your cells. Before we get into it, mitosis, again, as we know, that's cell division. There are these certain little checkpoints in mitosis, and we're going to talk about those in our next unit. But these little checkpoints, if they go unchecked, your cells grow and grow and duplicate and grow and duplicate and grow and duplicate. Well, they're not supposed to constantly do that. When they do constantly do that, that's what we call cancer when your cells constantly replicate and duplicate without any checks and balances. They just keep on doing it. And again, we're going to talk about that soon. But today we're going to just look at normal cell replication. Okay, the first stage of mitosis is called prophase. It's the first stage of mitosis. It's the longest stage. We have these little things called chromatin. They tighten and condense into chromosomes. Sister chromatids are the structures that contain identical copies of DNA. The sister chromatids are attached at what we call the centromere. So it's like the center part of the chromosome. So if you look at a chromosome and think of it like an X, each arm of the X is called a chromatid, and imagine a belt around the middle. That little belt is called a centromere. So as prophase continues, the spindle fibers, centrioles, and aster fibers form what's called a spindle apparatus. And the spindle apparatus attaches to each of the sister chromatids before cell division. So again, you have your chromatids attached by a centromere, making a chromosome. The spindle fibers attached to them, getting ready to pull them apart, basically. During metaphase, which is the second stage of mitosis, and it's actually the shortest phase of mitosis, sister chromatids are pulled along these spindle fibers from this spindle apparatus towards the center of the cell, and they line up in the middle of the cell here. And we actually call this this organized line of chromosomes, we call that the metaphase plate. Okay, so that's metaphase. Anaphase is when the sister chromatids are pulled apart. The microtubules of the spindle apparatus begin to shorten, so these little tubules shorten, which pull the chromatids apart and pull them towards the opposite poles of the cell. The chromatids separate and move towards each end here. And then we close with telophase, where the chromosomes arrive at the opposite poles of the cell. They begin to decondense, turning back into chromatids. And you have two new nuclear membranes that begin to form, and then the nucleoli reappear. The spindle apparatus goes away, it disassembles. And that's the end of mitosis. You now have these two complete nuclei, and you can tell they're not quite separated fully yet, but then we hop over to the final stage of the cell cycle, cytokinesis, and after those two daughter nuclei have fully formed, cytokinesis happens, which kind of pinches the cell in two, basically. So in animal cells, these microfilaments constrict or pinch to form two cells, and in plant cells, instead of pinching them off in half, a cell plate forms between them. Plant cells are a little bit different, but for our cells, they pinch apart. So we've had interphase, and then mitosis, and now finally cytokinesis. And once you've pinched off these two brand new daughter cells, you have completed a whole cell cycle. And then it's just going to start all over again until all of your cells get to the point where they need to duplicate. And then it starts over again. Then they live their life. Then they get to the point where they need to duplicate, and it starts all over again. You're probably starting to see why it's called the cell cycle, because it happens over and over and over again. And like I said earlier, we're going to talk more about what happens when that growth and reproduction gets unchecked next time. So hold that thought. If you need this information, watch this video as many times as you need to help yourself understand. Read the textbook if you need to. The information in your ed puzzles as well as the assignment are very important. And next week, we'll talk about just the next thing, what happens when it goes unchecked. Good luck on your assignments this week. And if you have any questions, make sure you let me know.